All right, let's dive into electroencephalography, or as you'll always hear it called, EEG. For almost 100 years now, this amazing technology has been our main window into the brain's electrical symphony. We use it everywhere, from deep research labs to the patient's bedside. So if you're a trainee in neurology or neuroscience, getting a handle on EEG isn't just a nice to have, it's absolutely essential. Let's get right into it.
So, to really get our heads around what EEG actually measures, let's start with a simple analogy. Imagine you're trying to report on a huge soccer match, right? But you're stuck way up on a hotel balcony, far, far away from the stadium. You can't hear what any single player is yelling, but you can definitely hear the collective roar of the crowd.
Okay, now, what if you get a little closer? Let's say you make it into the press box. You still can't hear the coach on the field, but you're starting to pick up on the chatter from the other reporters, the general hum of the audience right outside. This is kind of like recording what we call local field potentials. It's a more localized signal, but it's still a group activity.
And finally, if you got right down there on the field, stuck a microphone in the coach's face, you'd get incredible detail from that one person, but you'd completely miss the context of the overall game. This is what it's like to record the action potentials of a single neuron. Super precise, but you lose the big picture.
And that's EEG. It's that far-away measurement. It was never designed to listen to single neurons. Instead, its whole purpose is to capture the synchronized electrical buzz of millions and millions of them firing all at once. That joyful cry of the crowd when a goal is scored. It's the ultimate big-picture view of brain function.
So here's our game plan for this explainer. First, we're going to get crystal clear on what EEG can and can't do, hearing the brain's crowd. Then we'll dig into the neuronal source code to really understand the biophysics. After that, we'll take a trip through a century of innovation, figure out how we decode the brainwaves, and finally, see how EEG is being used in practice today.
All right, part one, hearing the brain's crowd. This is the absolute fundamental concept you've got to lock in. EEG gives us this amazing, real-time look at the brain's collective activity. Just think of it as listening to the synchronized hum of huge groups of neurons, not the individual chatter of single cells. Let's unpack what that really means.
Okay, moving on to part two, the neuronal source code. So we know EEG measures a group signal, but where does that signal actually come from? I mean, what's happening at the cellular level? Understanding the biophysics here is the key to being able to correctly interpret what you're seeing on that screen during a recording.
Now, this next point is absolutely crucial. If you're a trainee, this is one of the most important takeaways. When you look at an EEG readout, you are not seeing those fast all-or-nothing spikes from individual neurons firing. What you're actually measuring is the combined effect of much slower, smaller currents called postsynaptic potentials. These are the little voltage changes happening in a neuron when it gets a message from its neighbors. It's the sum of these tiny little potentials from millions of cells that finally gets big enough for us to measure on the scalp.
So for a signal to actually be strong enough for us to detect it, three things need to happen. First, like we said, huge populations of neurons have to be active in sync all at the same time. Second, the signal mostly comes from a very specific type of cell, the pyramidal neurons. Their shape and the way they're all lined up in parallel creates a measurable electrical field, kind of like a tiny battery with a plus and minus end. And third, that combined electrical current has to travel all the way from the cortex, through the brain tissue, the skull, and the skin to even reach our electrodes. It's a long journey.
Now that we know what we're measuring, let's take a look at the incredible history of the technology itself. You know, EEG might be an old dog, but believe me, it is constantly learning some amazing new tricks.
I mean, it's had a really remarkable journey. It all started back in the 1920s with Hans Berger, who, by the way, was pretty much laughed at by the scientific community at first. Then you have these huge leaps like event-related potentials in the 60s, all the way to today, where we're seeing brain-controlled therapies and real-time decoding. It's wild.
And wow, this slide just says it all, doesn't it? We've gone from these gigantic, room-sized analog machines that printed on paper to these modern, high-density systems that you can carry around and use right at the bedside or even out on a basketball court. The progress is just incredible.
And here's one of the main reasons it's still so critical in medicine. It's incredibly cost-effective. A clinical EEG system costs a tiny fraction of what an MRI or an MEG machine costs. And that makes this powerful window into brain function accessible to pretty much any hospital or a clinic anywhere.
Okay, on to our next section, decoding the brain waves. So we've put the electrodes on, we've recorded the signals, And now we're staring at all these wavy lines on a screen. How in the world do we make sense of them? Well, this is where the real ardent science of EEG begins, and it comes with a pretty big challenge.
The single biggest conceptual hurdle you'll face when you're learning to interpret EEG is something called the reference problem. And the best way to understand it is to start by asking a really simple geography question. How do you measure the height of a mountain?
You'd probably say, well, relative to sea level. Of course. But what if there wasn't a constant sea level? That's the problem with EEG. It measures a voltage difference between 2 points. But here's the kicker. There's no perfectly neutral zero voltage spot on the human body. So our sea level is always kind of arbitrary. And if you change your reference point, you can completely change how the brainwaves look, which makes interpretation really tricky.
So how do we get around this?
Well, here's the clever solution. While the specific voltage values might change depending on your reference, the overall shape of the electrical field across the whole scalp, the topography, that stays the same. The relationship between the peaks and the valleys doesn't change. So by analyzing this global map, we can get objective results that don't depend on the reference.
So, a modern analysis workflow looks something like this. First, we record the raw EEG data. Then we clean it up, filtering out stuff like the hum from the power outlets. Then we have to remove biological noise like eye blinks or muscle tension. The really key step is next. We chop up the data around specific events, like when a patient sees a photo, and we average them together. That averaging gets rid of the random noise and reveals the brain's consistent response, creating what we call an event-related potential, or ERP, and that's what we can analyze.
All right, let's wrap this up by looking at why all this matters in the clinic today. EEG isn't just for diagnosing epilepsy anymore. Far from it. It's actually powering some of the most exciting new frontiers in all of neuroscience.
I mean, the applications today are just mind-blowing. We're seeing brain-computer interfaces that let paralyzed patients control a wheelchair or type just by thinking. We have brain-triggered therapies that are helping stroke patients get motor function back years after their injury. And with machine learning, we're now starting to decode mental states in real time. It's just opening up a whole new world.
So yeah, far from being some dusty old technology, EEG is having a complete renaissance. Its amazing time resolution, its portability, its safety, it all makes it the perfect tool for the modern focus on brain dynamics and what's happening in the real world.
And this brings us to one last, really fascinating question. As our computers and our algorithms get exponentially more powerful, we are getting closer and closer to decoding the brain's signals in real time. This isn't science fiction anymore, so it leaves us to wonder, what's the next thought we'll be able to see?